Who am I? Where do I belong? And what is my potential? These questions might sound a bit too familiar in a room full of PhD students. And while these might sound like the questions I'm just asking myself right now, they're actually the questions I'm asking in my project about a population of vascular stem cells. Vascular stem cells, or VSCs for short, are the cells responsible for forming and repairing our blood vessels. If you take a second to think about all of the roles that our blood vessels play in our body, from providing blood flow to our organs and limbs, to allowing cancerous tumors to metastasize, you can quickly see how the ability to alter their formation and repair would be incredibly useful to the medical field. Despite the huge role that VSCs play in our health, they remain quite poorly understood, making them difficult to target in therapeutics. In my project, I aim to reach a more comprehensive understanding of this population in order to solve this problem. Let's start with our first question. Who is the vascular stem cell? These cells, much like PhD students, notoriously lack a uniform identity. They've been characterized across the field in numerous ways, all with distinct sets of markers. This confusion has led to several other types of cells being misidentified as VSCs, which has created further delays in targeting these cells for therapeutics. In my project, to address this, I have characterized our cells using several of the markers from across the field in order to try to find the overlap of these populations. Beyond this, I've worked to expand the definition of these cells based on their functional stem cell abilities, like the ability to create an entire population of cells from just one starting cell. This brings us to our second question. Where do vascular stem cells belong? It's not currently known where these cells sit within blood vessels. So far, I've found that in the aorta, there are specific regions where these cells sit when there's no injury or harm present. This is important because if we can more easily locate VSCs, we can more easily target them as well. Finally, what is the vascular stem cell's potential? Preliminary research has shown that these cells possess qualities and markers of both blood vessel forming cells and fibrotic cells that turn into muscle and connective tissue. This has led to the hypothesis that these cells may be bipotent, meaning that they're able to turn into both types of cells. If this is true, understanding the mechanism behind this could allow us to control how these cells develop. In the examples I provided earlier, we could favor fibrotic cells when we want to limit blood vessel growth, like in tumor metastasis, or we could favor blood vessel forming cells in cases like loss of blood flow. Throughout the rest of my project, I hope to uncover the identity, location, and potential of this population of vascular stem cells, and hopefully answer some of these same questions about myself along the way. Thank you.